So far we've been talking about how gene flow, drift, and adaptation act in isolation, but really all of these processes are interacting with each other. And so what we're going to focus on in this video is talking about how those different interactions can occur and how we know which one of those processes is going to have a stronger effect over another given a population's unique history. So the first two things we will talk about to compare are gene flow and genetic drift. So first of all, just as a quick refresher, go ahead and jot down or think about what are the definitions for these two processes, what do they mean, what kind of effects do they have. So what do you know about these two processes already? All right, so if we think about two different populations that have no gene flow occurring, what's going to happen is those populations are going to diverge from each other. They're going to become more genetically different as a result of drift. And remember, drift is due to chance, right? So that was the example I showed with poor Mustafa being trampled by the wildebeest. So if we have isolated populations, so say we have population 1 and population 2, and they're isolated and there's no gene flow occurring between them. What's going to happen is these isolated populations overall are going to have a lower effective population size. They're going to have a higher effect of genetic drift, and they're going to have lower genetic variation. And that again is because if you do have gene flow happening, you're bringing in new individuals with new alleles and new genes. But if you have isolated populations, that can't happen. So in isolated populations, drift is going to have a really strong effect. Your populations are going to differentiate just due to chance. You're going to have lower effective population sizes, higher effects of drift, and overall lower genetic differentiation in these isolated populations. And this is a really nice table that for some reason your textbook took out in this version. This was in previous versions of your textbook, and I put it in here because I think it's a really nice way to summarize the different effects. So if you, in these columns, we have two different types of populations that are connected by gene flow or isolated with no gene flow. And then we have different population genetics parameters, effective population size, genetic drift, genetic diversity, and population differentiation. So go ahead, pause this video, take some time to think about for each of these parameters, just think about a high arrow or a low arrow. So for effective population size, if you have connected populations with gene flow, is effective population size high or low? And just go ahead and fill in all of those different arrows. So go ahead and do that. This is your last warning to pause the video because I'm going to go over the answers now. So if we walk through this, if we first look at effective population size, if we have gene flow happening, we're going to have a higher effective population size. With isolated populations, we're going to have a lower effective population size. For genetic drift, if we have connected populations with a lot of gene flow, genetic drift is going to have a small effect versus in isolated populations, drift is going to have a really large effect. For genetic diversity, if we have connected populations where gene flow is happening, we're bringing in new alleles, our genetic diversity is going to be high. Versus isolated populations with no gene flow, our genetic diversity is going to be low. And then lastly, for population differentiation, if we have connected populations with a lot of gene flow, those two populations are going to tend to become more similar to each other. So our population differentiation is going to be low. If we have isolated populations with no gene flow, that differentiation is going to be high. So this is a nice summary of thinking about those two different competing scenarios and how each might have an effect on drift, effective population size, etc. The next two processes we're going to think about and compare are local adaptation and gene flow. Local adaptation we're going to spend an entire lecture on in a little bit. That's one of my favorite topics, so I'm going to dedicate a whole lecture to it, but we'll talk about it just briefly and introduce it here. The overall idea of local adaptation is that you have these environments that are different, they're heterogeneous. And so what that means is you're going to have different genotypes selected in different parts of that species range, and that's what leads to this local adaptation, or this fine-tuning of individuals to their local habitat through this process of natural selection. So when we think about the type of factors that make environments different, it's what we've talked about before. It can be an abiotic factor like temperature, which is shown here along the coast of Southern California. There are different sea surface temperatures. It can be salinity if we're still talking about marine environments, all kinds of different potential environmental factors that can cause adaptation for individuals living in different possible habitats, different parts of that range. And so the conundrum comes because you have these different populations. So this population lives in pretty cool water, this population lives in warm water. They're, through this process of local adaptation, going to have these unique genotypes that help them survive in different habitats. But if you have gene flow happening between them, like we just talked about, that's going to make those populations more similar. So one of the big questions in molecular ecology is, how do you have gene flow and also have local adaptation? How does that work? Because in general, gene flow is going to erase local adaptation by making two populations more similar to each other. And that really depends on the strength of selective pressure. And what that means is you can still have local adaptations happening even if you have a really high level of gene flow, as long as the selection pressure is really strong. It talks more about this in your textbook. I skipped it on purpose because I don't think it's super important for you to know how to do those types of calculations. But the overall idea is illustrated in this example from a recent publication. They were looking at different populations of deer mice in the Nebraska sandhills. And if you look at these two different little mice, they're the same species, but they live in different habitats in Nebraska. The one on the top lives in grassland, the one on the bottom lives in these sand dunes. And you can see that they also have different coloration patterns, and that's because their coloration patterns are matched specifically to help them blend in their unique habitats. There's one particular gene called the agouti gene that's responsible for these different fur color differences. And what they found overall when they looked at something crazy like 660,000 SNPs in this particular species, overall they found that there was very little differentiation. So overall across the genome it was very, very similar, which indicates there are high levels of gene flow. However, if you look particularly at that agouti gene, there are differences there that help to maintain this different cryptic coloration. So this is an example of how you can still have local adaptation, you can still have differences at that agouti gene, even though overall there are high levels of gene flow among other geographic sites. And then the last competing processes we'll talk about here are genetic drift and selection. So we're thinking about how drift and selection interact. And when we think about this, both processes cause populations to differentiate. So both drift and selection over time will cause two different populations to become genetically different from each other. The difference is that drift causes changes that are just due to chance, and selection causes changes that are actually adaptive. So how do we know which one of these processes is going to actually have a larger effect in a population? Well, the main factor is population size. Remember, as we've talked about previously, drift has a much larger effect in smaller populations. So, again, the main factor is population size. If selection is strong relative to the population size, then the effects of drift are going to be really small. And the converse is true. If the population is small, drift will have a larger effect on allele frequency changes. So say we have population 1 that's relatively small and population 2 that has a larger population size. In this small population here, drift is going to have a really big effect and will have a strong influence on population differences, and selection will have a small effect. And the converse is true here. For a population with a large population size, we're going to have a stronger effect of selection. Sorry, somehow I accidentally stopped my recording. I don't know if that's going to come out a little funky. And drift will have a smaller effect. And it's important for us to think about whether drift or selection is causing population differentiation, because it can have conservation management decisions. So say we have two different populations of butterflies, population 1 and population 2. If something happens and this population dies out, we want to potentially move individuals from another site into this site to help rebuild that population. However, if those two populations are differentiated due to selection, it might be that the individuals who lived here are only able to survive here, and the individuals who live here are only able to survive there. That was actually the case, and what happened, so in the Netherlands, they had populations of butterflies that were dying out, so they tried to move individuals from Belgium over to that site. That didn't work because the Netherlands population had really low quality plants, and only the butterflies who were from the Netherlands were adapted to survive on those lower quality plants. When they tried to bring in butterflies from Belgium, they couldn't survive because they didn't have the right food source. So this is an example of knowing that these populations are differentiated due to selection will help us determine that it does not make sense to move individuals in from a different geographic site. It won't be helpful because they won't be adapted to be able to survive in that new site.